I'd like to thank the Simons Foundation staff for inviting me up here as well. So I'd like to begin with this slide. This is one that highlights the autism prevalence in the United States. This takes advantage of CDC data as well as data from other sources. As you can clearly see, the prevalence has dramatically increased in the United States as it has in other countries. The current estimate is that 1 in 68 individuals has some form of autism. I think you all are familiar with what autism is. It's a neurodevelopmental disorder. And at its core, it has symptoms that include communication deficits as well as repetitive and restricted behavioral deficits. There's been a lot of talk as to what is leading to this increase. And I think to a large extent, this has to do with changing or broadening diagnostic criteria as well as increased awareness. We also know that there's a strong sex bias. There are roughly 4.5 times as many boys or males who are diagnosed with autism as opposed to females. And so there seems to be something about the female genome that is protective. And so in an effort to try and understand what is really driving autism or causing autism, it's become clear that there really are 2 key factors, genetic factors as well as environmental factors. The genetics we know much more about from heritability studies. And so these are studies looking at identical twins and looking at concordance, like whether or not the 2 twins both have autism or if only one has autism. And these studies point to a heritability of around 50%. And so that means that the chances of developing autism of the other twin is half. So there's a strong genetic component. And so the question is, where is this heritability coming from? So there was a really nice study done by Joe Buxbaum a few years ago, and pointing to the fact that a lot of this inherited risk is coming from common variants. So these are SNPs, single nucleotide variants, spread throughout all of our genomes that really make each of us unique. And there also are a small number of rare inherited variants that are giving rise to this risk. This is sometimes a difficult concept to understand, like this common variation and how that influences risk. There was actually a really nice interview by the spectrum of Thomas Bougeron, who's in France, who studies common variation in autism risk. And he came up with this analogy of driving through the hills or a canyon in Los Angeles in the summer, where everything is dry. And if a match got thrown out of your car, there would be a high probability that that match would lead to a fire. Whereas if you were to drive through that same canyon right now, which is rainy season, and I'm sure you all have been hearing about the massive amounts of rain in California, that same match would really have probably no effect whatsoever. So you can think of these common variants as either increasing or decreasing the propensity for something else, that match, to set off the fire, or in this case, autism. And so there's been a lot of work from efforts funded by the Simons Foundation to identify what some of these matches might be. And there's been a lot of effort placed on de novo mutations. These are new mutations that arise in a child with autism that aren't present in the parents. And so you can think of these de novo mutations like a match, essentially pushing a genome over the edge. Some of them are highly penetrant. The de novo mutation itself can give rise to autism. Others have variable penetrance. And so this is sort of the context in which our lab thinks about the environment. There clearly are some epidemiological studies linking environmental risks to autism, and I'm going to go over a few of those studies a little bit later. And so you can think of these environmental risks not necessarily as causing autism, but perhaps pushing a genome or pushing an individual over to this higher risk category, similar to a de novo mutation. And so I just want to focus briefly on these de novo mutations because a revolution has really taken place over the last 5 or so years in the area of autism genetics. And much of that has been funded by the Simons Foundation and the NIH. And so the gist of these studies is to take families, sequence unaffected individuals, as well as an affected child with autism, to try and identify a mutation that's present in the proband with autism that's not present in the parents. And from these exome sequencing studies, literally hundreds, if not thousands, of de novo mutations have been identified that that correlate with autism risk. And so I've listed just a few of these genes up here. So one of the top candidates is CHD8. This is a chromodomain helicase. And data suggests that about 0.43% of all individuals with autism will have a mutation in this gene. So that's 1 out of every 200 individuals. And so individually, each of these gene mutations is incredibly rare, but collectively they give rise to the spectrum that we see in the area of autism. And obviously, since I'm here, I'm not going to talk much more about these. My real interest is in environment and the question as to whether or not environmental risks might work similar to these de novo mutations and sort of push an individual or push a genome over to a high-risk category or one where autism develops. And I would also submit here that this represents one of the major biomedical challenges of our time. We currently have a very difficult time identifying environmental threats to the brain before they actually cause disease. And I also would like to point out that unlike these de novo mutations, which can't be avoided, environmental risks, once you know what they are, have the potential of being, you have the potential to avoid them, especially during critical periods of brain development. So if you can identify these risks, there's the potential to at least eliminate an avoidable, what I would consider an avoidable form of autism. We're not going to be able to eliminate de novo mutations, but at least for a subset of autism cases, identifying environmental risks may allow us to reduce the prevalence to some extent. And so the approach that our lab has taken is one that I call a prospective approach. And the general idea, it's pretty simple. If we have these de novo mutations that are individually linked to autism risk, can we find chemicals that target some of the same molecular pathways that these de novo gene mutations are located in? And so if a de novo mutation in CHD8 or Nerexin-1 can push an individual over to develop autism, could a chemical that targets that same molecular pathway do the same thing? And if so, how do we find those chemicals? And so thanks to these studies of de novo mutations, we now have this long list, a relatively long list of genes linked to autism risk. And a number of labs have done a number of analyses to try and assess whether or not autism is incredibly heterogeneous or if these genes cluster in certain functional pathways. And what has come about from a number of these studies is that this is showing a relatively recent study looking at maybe 9 different functional pathways, but many of these analyses tend to center on a much smaller number of pathways linked to synaptic function, as well as to early developmental effects in the Wnt beta-catenin pathway. And then there's also the C4 module. A lot of these autism-linked genes are linked to transcriptional regulation and the immune response. And so I'm going to highlight how environmental risks that target these pathways could be identified. And so as far as synaptic function is concerned, as many of you know, the synapse represents the point of communication between neurons. I'm going to show a blow-up of a synapse next. So a synapse is made up of a presynaptic neuron that dumps neurotransmitter into the synaptic cleft and is received by the postsynaptic neuron. And what I've circled here are a number of genes that build the synapse and that are also linked to autism risk. Several years ago, our lab made the observation that many of these synaptic genes have a unique physical property, and that is they are unusually long. They take up a lot of space in the genome. And so, for example, these autism genes are all well over 100, many of these genes are well over 100 kilobases, and I highlight one gene, Nerexin-3, as an example. This is a gene that is 1,612 kilobases, that's 1.2 megabases. And so I drew this to scale relative to the average gene, which is about 60 kilobases. And so many of these synaptic genes are incredibly long, 20 times longer than the average gene. A few years ago, I wrote a viewpoint for Safari about what does it mean to be a long gene, what are the biological implications. So a big gene, for example, is going to be a bigger target for random mutations. Big genes take a long time to transcribe. An RNA polymerase starts at the beginning of a gene and has to go all the way to the end before a transcript is produced. It can take anywhere from 6 to 10 hours to get from one end of a big gene to the other, whereas a short gene, maybe 10 minutes. And so chemicals that perturb this transcription process have the potential to disproportionately affect the expression of long genes and proteins relative to shorter genes. And the other thing that is interesting about long genes, when you sort of think about it, or when you look at it, when you look at gene expression through this length perspective, is that the nervous system seems to be unique. What we're looking at here is expression data from a number of mouse tissues, ranging from the amygdala all the way to snout epidermis. And what we did is we highlighted all of the neural tissues in red and the non-neural tissues in gray. And then we looked at the gene expression data from each of these tissues and generated a single number that represents whether or not the expression of all the genes is biased. So are there relatively more long genes in this tissue or relatively more short genes expressed? And if the number is positive, this is what we call a positive length correlation, that means there's an overrepresentation of long genes in that tissue. And what you can clearly see is that there's an overrepresentation of long genes in neural tissues relative to non-neural tissues. Yep. Did you have a question?
Do you find the mutations occur at the same spot in the gene?
These de novo mutations in general are all over the genes. Should we wait till the end for questions? And so then the question, so these are tissues like amygdala, frontal cortex, and the brain obviously is made up of many cell types. And so the question is, what cells are driving this length correlation? And so with the advent of single cell sequencing, we were able to go back and look at some of this data, and we find it's the neurons that overexpress these very long synaptic genes. Other cells like astrocytes and ependymal cells tend to not overexpress long genes. And so then this now raises the question, like if there are chemicals that affect disproportionately long synaptic genes, do they exist, and might they affect synaptic function? And so a few years ago, we made a novel observation regarding a class of chemicals called topoisomerase inhibitors. These are drugs that are used to treat cancer. We made this observation because we had been studying topoisomerase inhibitors in the context of a different neurodevelopmental disorder called Angelman syndrome. I don't have time to go over that study. Much of it has been published. But suffice it to say, as we drilled down to try and understand better how topoisomerase inhibitors worked at the gene expression level, we found they had a disproportionate effect on the expression of very long transcripts. And so what you're looking at here is gene expression comparing drug-treated cells to vehicle. And then along the x-axis is gene length. Each dot represents the average expression change of 200 genes binned by length. And so this helps clean up the data. And as you can see, shorter genes are maybe increased a little bit in expression. Once you get to about 100 kilobases, you see this dramatic reduction in expression. And when we looked at what those long genes were, as I mentioned, they were all primarily long synaptic genes. And so this has implications at the protein level. So Nerexin-1, NRX1, is an high-confidence autism candidate. This is now the protein level. If you look at the leftmost lane, you can see that prior to drug treatment, there's plenty of protein in these neuronal cultures. After treating with Topotecan for 72 hours, not only does it downregulate long genes, but it also virtually knocks out the protein. This effect can be washed out. As you can see, 24 through 72 hours of washout, the protein bounces back. Another long gene linked to autism is neuroligand, which is the ligand for Nerexin-1. They interact. That also is downregulated in a reversible manner. And so you can begin to see how a chemical has the potential to target many of these long genes, many of which are linked to autism risk. And so we also wanted to know whether or not these chemicals affect synaptic function. And so we did a number of electrophysiological experiments, all of which are published. I just highlight one experiment here where we looked at network activity in cortical neuron cultures. And so if you put cortical neurons in a dish and let them grow for several days, they eventually form synapses and start to fire in bursts. And you can see these bursts electrophysiologically, and you can also see them using a calcium-sensitive dye. And so that's what we're looking at here. These are neurons loaded with Fura2, calcium-sensitive dye, and then we just imaged over time, and you can see these bursts. Each of those lines is a different color that represents a different neuron. You can see each neuron is bursting in synchrony at a low amplitude. When we add this drug, GBZ, that's gabazine, that increases the amplitude of that bursting. But you can see that they still are all bursting in synchrony. And so this is a measure of synaptic activity, network activity in culture. And we know that it's glutamate mediated because all the way over there at the right, DNQX is a glutamate receptor blocker. So we can eliminate this bursting with DNQX. When we do these same experiments in Topotecan treated cultures, not only are the long genes down, including a lot of these synaptic genes, but it also virtually eliminates this spontaneous network activity in culture. And adding gabazine really has no effect whatsoever. So you see this profound reduction in synaptic activity in the presence of this drug that downregulates long genes. And so are there other chemicals or other genes linked to autism that might work through a similar mechanism? And so when looking at the de novo data, the answer is yes. And so there's another gene, high confidence gene linked to autism called SCN2A. It's a voltage-gated sodium channel. There are a number of missense mutations in this channel. So missense mutations are mutations that change an amino acid. And many of these mutations are located very close to the ion selectivity pore. There was a recent paper that came out from Kevin Bender's lab just a few weeks ago, actually maybe a week or two ago, trying to understand how these mutations affect channel function. And basically what his lab found is that these autism-linked mutations in this channel seem to dampen or eliminate channel function. And so this is interesting because there is a class of environmental use chemicals that are very common. They're called pyrethroids, which also target voltage-gated sodium channels, including this one. They essentially kill bugs by binding to the sodium channels in the bug brain, but these pyrethroids also interact with mammalian sodium channels. And so these pyrethroids are present on our food. They're used to eliminate pests. They're also found in our houses quite commonly. So when an exterminator comes in to treat for bugs, pyrethroids are typically used. And they appear to be present in humans of all ages. There's a study run by NHANES that samples human blood and urine of children of all ages, and they find that virtually all of the people sampled have detectable levels of pyrethroids or metabolites in their body. So humans are definitely exposed to this class of chemicals. And so at a neurobiological level, these chemicals prolong the open state of these channels, and then when the channels close or inactivate, it also prolongs the closed state. So these channels have the potential to essentially ultimately and long-term shut down the channels. We know that acutely, these pyrethroids, when added to mammalian neurons, will cause immediate early gene induction. And so this is a transcriptional readout that I'm going to come back to. We also know from using similar experiments that we did with calcium imaging that they blunt synaptic activity, much like Topotecan. And so this is work done by Tim Schafer at the EPA, which is actually right down the road from UNC. So these are two different pyrethroids showing dose-dependent reductions in spontaneous network activity in culture. So these chemicals seem to have functions they target an autism, high-confidence autism gene and have functions that are very similar to autism-linked mutations in that gene. And perhaps most relevant of all, epidemiological studies have recently linked pyrethroid exposure during the third trimester to increased autism risk. And so this was a really nice study where it was done in California. California has really good environmental sampling data at a regional level. And so they went in and they were able to assess exposure levels to a variety of pesticides of pregnant women and look at the proximity of exposure to autism risk. And they basically find that the closer a woman is to an exposed area, the greater their risk for autism, for having a child with autism. And so then this raises the question, could pyrethroids or chemicals that act like pyrethroids that are present in the environment that humans are exposed to represent chemical risks for autism? Could they essentially flip the switch similar to a de novo mutation in a channel like SCN2A? So I also talked about the Wnt beta-catenin signaling pathway, as that's another high pathway that seems to be implicated in autism. And so for those of you not familiar with this pathway, at its core is a protein called beta-catenin. This is a protein that is held in a complex called the destruction complex. And while in that complex, it is phosphorylated by a protein called GSK3-beta. And when phosphorylated, it is targeted for degradation by the proteasome. So this destruction complex essentially eliminates beta-catenin. in the presence of Wnt ligand. Wnt is a protein, it's a secreted protein. That leads to the dissolution of this destruction complex, and beta-catenin is then free to enter the nucleus and interact with transcriptional regulators, such as CHD8, to regulate Wnt target genes. And so this Wnt pathway has been heavily studied in the context of cancer. There are a number of mutations in this pathway that lead to overproliferation of cells and cancer. But we're now realizing, thanks to work from the Simon's simplex collection sequencing and others, that a disproportionate number of autism genes are in this Wnt signaling pathway. So Alan Packer wrote a recent editorial about this. At least 16% of all the ASD genes are are in this pathway in some way or another. And so that includes this gene, which I keep talking about, CHD8. This is one of the highest confidence autism genes. Macrocephaly, or an enlarged head, is one of the common phenotypes. Over here on the right is the growth curve of head circumference, tracking 2 individuals. And so they're on the upper end of the growth circumference curve for children. And so this overgrowth, this cortical overgrowth, is interesting for another reason, because many labs, not many labs, but Joe Piven's lab, as well as Eric Corshane's lab, have noted that cortical overgrowth is a relatively common phenotype in many individuals with autism. And so just to make a plug for Joe's work, just last week he had a paper published in Nature, and using brain imaging data, he was able to show that you can predict whether or not a child was going to go on to develop autism based on a brain scan between the ages of 6 months and 12 months of age. And so there are changes in the brain enlargement, that precede the diagnosis, behavioral diagnosis of autism. So this really highlights further the idea that autism pathogenesis really initiates very early, probably prenatally, and overgrowth is one of the common phenotypes possibly related to this pathway. So if this pathway is implicated, are there chemicals that target this pathway that are also linked to autism? And so the answer is yes. And so there's a drug called valproate, or valproic acid. This is an anti-epileptic. This is a drug that's used to treat epilepsy in humans. It works quite well. It's also known that valproate, when given to women during pregnancy, can increase the risk for birth defects. But despite that risk, women who have epilepsy often still take this drug, because the risk of having a child with a birth defect is relatively minor compared to the risk of having a seizure and dying during pregnancy. So if valproate is good at treating her seizures, they'll often remain on this drug. And so for children who, so valproate is also really the model that everyone in the field uses as an environmental risk for autism. So valproate exposure in rats as well as in mice produced during the prenatal period leads to autism symptoms in the mice or the rats. And a very recent study has shown that exposure throughout the course of development in mice leads to cortical overgrowth. So this is another example of a chemical targeting the Wnt pathway, affecting brain development and linked to autism risk in rodents. But I think the key paper is this one published back in 2013, where they looked at mothers, children from mothers who took valproate during pregnancy, and found a significantly increased risk for autism in their offspring. And so I think this provides another good hint that chemicals that target the Wnt pathway, or chemicals that act like valproic acid, could represent environmental risks for autism. So the last pathway I just want to talk about is neuroimmune activation. And so it's also well known epidemiologically that prenatal, when mother infection during the 2nd trimester is also implicated in autism risk. So we call this the maternal immune activation model. You can mimic this in rodents by injecting poly-IC, which mimics this viral neuroimmune activation. And so also in postmortem studies of children and young adults, as well as older adults, looking at transcriptomic changes in the brains of individuals with autism, neuroimmune activation seems to be a prominent feature in many, but not all individuals. Now, this is really a landmark paper from Dan Geschwin's lab, where he transcriptionally profiled the brains of individuals with autism versus controls, and he found two classes of genes that seem to be misregulated. And so in many of the individuals, these red bars going down showed reduction in the expression of many of these synaptic genes, these long synaptic genes, and then elevated expression of neuroimmune type genes. A number of other studies, Dan Arking's lab has reproduced this. There's clear evidence of neuroimmune activation in older individuals with autism. And so might chemicals that target the neuroimmune system or lead to immune activation represent environmental risks. And so the general idea, I've highlighted a couple of pathways now strongly implicated in autism. And so the question is, can we identify environmental use chemicals that target any of these pathways? Synaptic function, we've given an example of topoisomerase inhibitors. Might there be other chemicals like topoisomerase inhibitors in the environment? Sodium channels, pyrethroids is one example, but there might be others. Wnt beta-catenin pathway, and then neuroinflammation. So the approach our lab has taken to identify these chemicals or these candidate risks is a transcriptomics-based approach. And so the idea is pretty simple. What we do is we take chemicals from, initially we're starting with the EPA's ToxCast library. This is a library of about 300 environmental use chemicals. So this includes pesticides and herbicides, plasticizers, et cetera. We add them one by one to cultured cortical neurons. We also used Topotecan and other topoisomerase inhibitors as positive controls and vehicle-treated negative controls. And then the question was, could we find chemicals that mimic some of these pathways that are affected by autism? Before doing this, we pre-screen the library for toxicity because there's obviously no point in sequencing dead neurons. And so we did this in 96 well dishes. We found a non-toxic dose, and that was the dose that we ultimately sequenced. And so this is relatively low throughput. The neurons were dosed in 12 well plates. RNA was collected, multiplexed 24 samples per lane, and then sequenced. Before I show you the data, I just want to highlight the utility of these cultures, these mouse embryonic cortical neuron cultures. They seem to, because ultimately our goal was to compare the transcriptional profiles in these cultures to human brain data. And so we wanted to make sure that these cultures transcriptionally modeled the human brain. And so using single cell data, we were able to confirm that these cultures have more or less all of the cells in the brain, neurons, glia, et cetera. We used immunostaining to also show that these cultures contain all of the cell types. And then we also used transcriptomics. And so what we did is we took RNA-seq data from these cultures and then compared that to RNA-seq data from many different human tissues, brain tissues in red, and then non-brain tissues in black. And as you can clearly see, the mouse data correlates best with human brain tissue as opposed to all other brain tissues, all other human tissues that are non-brain. So this contrasts with a different cell, which are human embryonic stem cell-derived neurons. And so many people are using human ES-derived neurons or IPS-derived neurons to study autism risk. But at least in this case, these human ES-derived neurons do not model the complexity of the human brain as well. And so what we did here is we took RNA-seq data from IPS-derived neurons and looked at the same GTEx data. And we find that there's very little correlation between ES-derived neurons and human brain regions. And we think this has to do with the fact that when you generate an ES-derived neuron, you generate a neuron. You don't generate the microglia and the astrocytes and all the different cell types that make up the brain. And so we think it's actually important to have these different cell types because the transcriptional changes that take place in the brain of autism aren't just neurons. It may be neurons and then the glial response to what happens in neurons. And so we did this experiment. This is now the summary of like 2.5 years of work and a lot of money on RNA-seq. Along the top axis, along the x-axis effectively, are the 300 plus chemicals. And then along the y-axis are the 5,000 variably expressed genes. So these are the genes that increased or decreased in some proportion of the samples. We basically threw out all the genes that were just not expressed because they really had conveyed no information. And we then performed batch correction and normalization of the data so we could combine all of these experiments that took place over the course of 2.5 years. And we were very happy to see, after clustering the data, so we were clustering to try and find chemicals that had similar transcriptional effects in these neurons. We were happy to see that the topoisomerase inhibitors that we use all clustered together. So we used 3 different topoisomerase inhibitors. And the main 2 effects that they had, one was downregulation of many long genes. And so we show the long genes are here, and we show that they're long by putting a tick mark next to any gene that is 100 kilobases or longer. So you can see a lot of the long genes are represented in this little patch. The other thing we noticed was that these topoisomerase inhibitors downregulated these genes. And if you look over here, those are all immediate early genes, like FOS, BDNF, et cetera. And so this is consistent with these chemicals downregulating long synaptic genes and dampening synaptic activity, which will have an effect on dampening immediate early genes, which are normally turned on when cultures are bursting. So I just want to briefly highlight cluster 1. I'm not going to talk more about it after this slide. But this cluster has a number of chemicals that include pyrethroids. And so you can clearly see that these chemicals are inducing immediate early genes. And remember, I said that acute application of these chemicals causes calcium influx and immediate early gene induction. And they also downregulate potassium channels. And so this is a unique transcriptional signature of this pyrethroid class of chemicals. The chemicals I want to talk about for the remainder of this presentation are the cluster 2 chemicals. So these are chemicals that upregulated, red, many neuroimmune genes, as well as cytoskeletal genes, and then downregulated a number of ion channels and some of the long synaptic genes. And so we tried to then assess whether or not these cluster 2 chemicals transcriptionally mimicked the changes that are seen in the brains of individuals with autism. And so to do that, we used a technique called gene set enrichment analysis. This is where you take the list of genes that are upregulated in autism brain or downregulated in autism brain and use a statistical test to see if your chemical class upregulates or downregulates those same genes. And so we did this for each of the clusters going across the top. And as you can see, the only chemical class that regulates autism genes, the only chemical class that causes this transcriptional signature of autism is cluster 2. And so if you look at the downregulated Gupta mod1, so these are the synaptic genes that are down, these chemicals downregulate those genes. And then the upregulated Gupta mod5, those are some of those neuroimmune genes. So they were significantly upregulated. We also looked at, in the Vonnegut data set, we saw the same thing. And so basically these chemicals are downregulating synaptic genes and upregulating neuroimmune genes, very similar to what is seen in autism brain. What was interesting is that this cluster 2 class of chemicals also look like other other human brain tissue, including aging brain and some forms of neurodegeneration. And so what we think this chemical class is picking up on is an underlying signature of neurodegeneration in the brains of at least some individuals with autism. My colleague Joe Piven has actually been trying to track down older individuals with autism just to find out what are some of the symptoms of older individuals with autism, and he's finding a significant number, like 1 out of every 4 have some sort of movement disorder or Parkinsonian-like condition. And relative to the general population, Parkinson's affects maybe 1 out of every 5,000. So there may be some underlying neurodegeneration that's taking place as individuals with autism age. And this transcriptomics signature might be picking that up. And so what are these chemicals in cluster 2? So 2 of the chemicals are shown here, phenamidone and praglistrobin. And these are chemicals you probably have never heard of. I know I never heard of them when I started this project. You can see that they don't look like one another. Structurally, they're very different. But both of them are known to be fungicides that target basically fungi that grow on plants. And so these are used to treat molds and rusts and things like that on food crops. And so we then did RNA-seq, more RNA-seq, just to assess at a really statistically significant level whether or not they up and down regulate the same genes. And so that's what these Venn diagrams are showing, is that even though they look structurally different, they are turning up and down more or less the same genes. And so some of the genes that they're affecting are shown here. This includes GST genes. These are glutathione transferase genes. These are genes that are induced in response to, typically induced in response to oxidative stress. And so this is qPCR validation that these genes are getting turned on. And then over in panel F, we were able to show that there's a dose-dependent increase in the expression of several of these genes. And so these chemicals, the way these chemicals work is by poisoning mitochondrial complex 3. So the mitochondria are the energy sources, generate energy for your cells. And if complex 1 or complex 3 are poisoned, superoxide, that's what the O2 is, builds up, and this can cause damage to neurons. And so this superoxide is also known as ROS, or reactive oxygen. And so we wanted to see if these chemicals were indeed inducing ROS in these neurons. And so we used a dye called mitosox, which turns red in the presence of reactive oxygen. So these are cortical neuron cultures, vehicle treated and phenamidone treated. And you can clearly see they turn red. They also sort of round up, and ultimately they would die if you left this chemical on long term. These are pretty nasty chemicals. And so we quantified both the ROS production and this change in cell morphology. And we found that both of them, the ROS and the morphological changes, were dose dependent. And then what I circled there in red are the doses that we used for sequencing. And so this transcriptional signature of autism is induced at drug concentrations that induce these reactive oxygen species changes as well as morphological changes. And so with ROS, one way that people often try and block this is with vitamin E, which is an antioxidant. And this is just showing that the drug causes ROS, and that if you pre-treat with vitamin E, you can block the ROS. You can also block the aberrant cell morphology with vitamin E. And what was interesting is since the cells change shape, we were wondering if there might be effects on the cytoskeleton. So we looked at actin and we looked at microtubules. And so it turns out that these chemicals are not only producing ROS, but also destabilizing microtubules. And so if you use a drug, Taxol, which stabilizes microtubules, that can reduce the formation of ROS and also reduce the aberrant cell morphology. And so these two processes, ROS and microtubule destabilization, seem to go hand in hand. And so this is great. This was RNA-seq. We did like 300 chemicals, but we ultimately really want to accelerate the pace at which we find these chemicals and also look beyond just cluster 2. Ultimately, there are 80,000 plus chemicals approved for use in the environment. Many of these, in fact, most of them, have not been tested on the developing brain or even adults. And so we have very little understanding of how these chemicals affect the developing or adult brain. The approach that we used, RNA-seq, is labor-intensive and costly. It costs us about $200 per chemical dose. And so we really wanted to find a cheaper way of doing this. The nice thing about the RNA-seq data is that it at least told us what genes best represent each of those clusters. And so we really don't need RNA-seq to find more chemicals in any of these clusters. Instead, we can use gene markers. And the approach that we took to do this was a relatively new high-throughput sequencing approach called RASL-seq. RASL-seq stands for RNA annealing, selection, and ligation. And so you can think of this as a quantitative PCR on steroids. It's a much more simplified procedure. You take neurons, you can put them in 384-well dishes, you lyse the cells, and then you can directly interrogate gene expression without using reverse transcriptase and cDNA construction. So you basically lyse the cells, you then add a magnetic oligo-DT labeled bead, and you fish out the polyadenylated transcripts, and then you add probes to whatever genes you're interested in interrogating, such as those GST genes. These probes will bind to the RNA template and hybridize. And if they hybridize, you then add a ligase, an RNA ligase, that will join those two pieces together and create a 40 nucleotide sequence. You then use PCR with unique barcodes that are specific to each plate and each well, amplify, and then sequence. And then you can basically combine all 384 wells or thousands of wells together in a single high-seq and do quantitative transcriptomics on hundreds of genes. And so the advantage of this is that it's incredibly high throughput. We've got this up and running in the lab. It's automated. We can process a single 384-well plate of neurons in under 3 hours. The barcode combination allows for 36,000 different combinations, so you can test 36,000 different chemicals if you have the money to culture 36,000 different wells of neurons. And this can be done for under $2 per replicate. So this is just showing what the data look like. So this is a reduced probe set now. This is a probe set that will interrogate different clusters. And so up top, you can see cluster 1, cyfluthrin is a pyrethroid at increasing doses. You can see that at increasing doses, it turns on some genes there, and they happen to be immediate early genes, but they don't consistently turn on anything else. And so we'll be able to identify pyrethroids based on their unique ability to induce immediate early genes in these cultures. Cluster 2 is phenamidone. As you increase the dose, you can see that that turns on a cluster of genes. And those are the genes that mark the neuroinflammatory cluster. They also downregulate some genes, some of the synaptic genes. And that's a unique transcriptional profile. And then the last is cluster 5, these topoisomerase inhibitors, which downregulate long genes. And the long synaptic gene cluster is down there at the bottom. And in previous work, we found that topoisomerase inhibitors also upregulate UB3A. So UB3A is on there, and you can see a dose-dependent increase in UB3A. And so we can use these unique transcriptional signatures in this automated manner to now scan through thousands of chemicals and use transcriptomics to prospectively identify candidate environmental risks for autism. And so we've essentially taken a technology that's typically used for drug discovery, and we're trying to now use it to find chemical risks for autism. So what are the next steps? And so obviously, when you're screening thousands of chemicals, we're going to find a number of things that fall within each of these clusters. But many of those are not going to represent risks, especially if humans are never exposed to them. And so one of the other things that we're very interested in doing and working on is trying to assess exposure threat to humans based on existing environmental sampling data, and then trying to generate our own environmental sampling data. And then the other thing is to then validate risk potential in vivo in animal models. So do the chemicals cause, quote unquote, autism in an animal model like valproic acid does? Or do they only do that in a genetically sensitized background? So like, for example, in an autism mutant mouse model. And so assessing exposure threats. So this just gives you an idea as these chemicals are applied to our food. The EPA and the USDA tracks pesticide residues on our food to make sure they're within allowable limits. These data are publicly available, and so we've gone through and analyzed all of that data for a number of cluster 2 fungicides. I'm just going to show you one here. This is pyraclostrobin. And so this is a chemical class that really was only recently approved for use in the environment. So pyraclostrobin was first approved in 2002. That's what that little red arrow indicates. And then what this is showing is how many millions of kilograms were used in the United States. And you can see there's an upward trajectory. And in fact, for all of these fungicides in cluster 2, their use is skyrocketing because they're very effective at treating fungus. And over here is a graph that just highlights some of the foods that contain these residues. This is looking at the maximal, the foods with the highest levels of ever detected of these chemicals. And so I think it's important to point out that this is just like the potential maximum. On average, these foods may have much lower levels or no levels. So just keep that in mind. Don't get too freaked out. You can still eat your spinach. And so to summarize this part, what we've been able to do using transcriptomics for cluster 2 anyhow, is to identify a class of chemicals that transcriptionally mimic the changes that are seen in the postmortem autism brain. We know from data that the EPA collected using something called reverse dosimetry, where they take those environmental sampling data and then extrapolate that to cell-based models, and then try and figure out whether or not the dose on the food has the potential to affect human biology. And so they found that for many of the chemicals, if there are only a small number of chemicals, there is this potential, and pyraclostrobin was one of them with the potential to affect human biology. These are foods, can be found in baby foods. So if there's anything with spinach that's not organic, it has the potential to have these chemicals. The good news is that you can avoid them, these foods, by eating organically. So this is sort of where we stood when we wrote this paper. I was obviously very interested in figuring out where else these chemicals might be found in the environment. So in doing more research, I ran across this patent that was submitted in 2006 from Syngenta. And if you read the yellow highlight, it says this invention relates to the treatment of wallboards with a fungicidally effective amount of strobilurin or strobilurin type fungicides, and with a focus in the next sentence on azoxystrobin, which is one of these chemicals that can induce this transcriptional signature. And so I then did a little bit more searching and found out that these wallboards are available now. You can buy them. They first started being marketed in 2009. I went to my local Lowe's, and sure enough, I took a picture. These are purple wallboards, and they're being used in homes these days. You can't read the sign. It says purple, but in small text, it says purple is the new green board. And so for those of you who know about building, like whenever there's an area that's sensitive to mold, like showers and kitchens, green boards are typically used to limit mold growth. But they're now marketing these for use in lieu of green boards. And in fact, I couldn't find green boards when I took this picture. But I think what may be more troubling is that they're also marketing these for use throughout the house because people are afraid of toxic molds and things like that. And so these are boards that are now being put literally everywhere. And this is the point where I want to also now highlight that in cluster 2 was another chemical called rotenone. Rotenone also poisons mitochondria, complex 1. That's a chemical that's epidemiologically linked to Parkinson's disease risk. And so rotenone has been largely phased out of use because of this linkage. And so we are now building houses with a chemical that basically targets the same molecular pathway. And essentially our whole houses are going to be potentially filled with low levels of these fungicides. So a future direction, working with other colleagues, is to start environmental sampling in homes and correlating levels of some of these fungicides with construction, new construction versus old construction, renovations, et cetera. And that's really just the first step to beginning epidemiological studies linking exposure to autism risk or neurodegeneration risk. And so the last step is to ultimately validate risk potential in vivo. And so we don't have final data on this. I just want to highlight the direction that we're trying to go with cluster 2 chemicals as well as other cluster chemicals. And so do the chemicals cause or exacerbate autism symptoms in wild-type or autism model mice? And so for the strobilurin fungicides, we know that in vitro they induce ROS, they destabilize microtubules, which can affect neuronal migration, and they cause this transcriptional signature of autism with neuroimmune genes up and synaptic genes down. And so as not to reinvent the wheel, we went and looked at a lot of the toxicological assays that were already done to try and figure out just what to dissolve these chemicals in. And this is where we got our first sort of surprise, I guess. Basically, all the tests that were used to evaluate whether or not these are toxic in mammals used aqueous solvents, specifically methylcellulose or CMC, which is carboxymethylcellulose. And as you can see, A and B, this stuff doesn't dissolve in these aqueous solvents, but it does dissolve very effectively in corn oil. So I'm not going to show you the data, but we basically did some in vivo studies in these different solvents, and we only really see effects in the corn oil, as you might expect, because the chemical doesn't even get in with methylcellulose. And so some of the effects we see, so we actually have gone down and lowered the dose because we're trying to find low doses and higher doses that are not lethal to the animals for these autism type studies. And so we found that the 3 samples are black is corn oil, which is just the vehicle, gray is corn oil and 10 mg per kg pyraclostrobin, and then red is a higher dose. And so you can see that as you increase the dose, one of the main symptoms is basically massive diarrhea. You also see a big change in body temperature that lasts for at least a day, reduction in body weight that lasts at least for 2 days at the higher dose, and then a reduction in motor activity. So we look at wheel running in the animals, and this causes the animals to just not want to run for some reason. And so now that we've identified doses, we can now go in and do studies like this, where we can look at acute exposure or chronic exposure during the window of of brain development, E11.5 through P0, where the brain, where the cortex is developing, and then assess at different time points, like at birth, 3 months, as well as 6 months, brain weight, as well as histological and transcriptomic phenotypes. So do these chemicals alter the size of the brain? Do they induce this neuroinflammatory signature? And do they cause behavioral phenotypes of autism? So I will end there and just acknowledge that this has been a collaborative project with many people in my lab. I see that I'm running out of time, and I want to make sure I have a chance to answer questions. So I'll leave it there and take your questions.